technology that we're going to look at, where it's actually sort of small dishes of analytes and molecules that we're going to address. And we call it the Swedish smörgåsbord, which is the small collection of dishes we have on the traditional table. So the dish I will introduce is called surströmming. It's a very, very odd thing, and it's something that I can't recommend you to test it, but maybe. See if this works. A little movie here about yes.
They say that when you first open a can of surströmming, it's one of the worst smells in the world.
Let's not do it then.
I don't want to.
Oh! Broken the seal.
Yeah.
Oh! I don't.
Oh, God.
Okay, nope.
I'm not.
Oh, God,
Oh my God.
no! It's sewage. It's sewage in a can.
Oh my God.
I'm out. Can we leave? Oh, it's coming. It's traveling. It's traveling.
Oh God. Someone turn on the air.
I have the worst gag reflex. I'm definitely going to heat.
It's one of the worst things I've ever smoked.
In order not to take too much time, I can recommend you to go and see this movie. It's on YouTube, but it's a very funny way of describing traditional Swedish food. I think after watching the movie, you want to go and buy this dish, I think. But there are things in here that causes problems. So we have, of course, things in our close environment and also around us that disturb neurotransmitters and immune-reactive molecules that can, of course, cause this delicate balance between the immune system and the central nervous system to be offset. So poor diet, surströmming, for instance, or toxic consumables that we approach sometimes, sensory overload, bowel dysfunction, genetics, of course, chronic stress, external stress, and then environmental toxins and things like that. So it all comes together and also make the situation very complex when we study it. This busy slide, unfortunately, I will try just to tell you a bit what can be examples of things what can happen between the central nervous system and the immune system. So if you see in the top panel, you see that we have the central stressors, those that affect the central nervous system directly. And it can be physical stress, it can be psychological stress, it can be immunological stress, or infections, for instance. And you see that the brain will react on different ways. It will start to produce neurotransmitters that actually then can be transported and affect, for instance, a down-regulation of the immune system, or sometimes an up-regulation of the immune system. And it can also go through the autonomic nervous system and control, for instance, our bowel function, motility, secretion in our gut. And that together with the peripheral stresses that could be an infection or inflammation comes together with the immune cell regulation. And it all becomes a very, very complex situation where inflammation, motility, secretion, regulatory mechanism in the central nervous system is affected. Just to complicate things a bit, many years ago we discovered in our work that our classical neurotransmitters, catecholamines, they actually are very important immunoregulatory molecules also. So dopamine, for instance, can regulate the immune system. And this little slide here just shows you what can happen. So if we have normal levels of catecholamines, the immune cells, this is a T cell, immune cell, they are fairly nicely regulated. They proliferate and they differentiate as they should. But if we have increased catecholamines in the region, actually we will affect the proliferation, differentiation, and also induce apoptosis, which is programmed cell death in the immune system. And it's not only external catecholamines, it's also internal. So the lymphocytes, they can produce their own catecholamines, and they have receptors for these molecules, and they also have transporters, so they can even transport these molecules into the cellular nuclei, which is then a possible way of action. So how can we do then to measure all these molecules to try to understand the system? Well, it's not trivial, but by selecting different targets, if we go for genetic analysis, we approach groups like Ron's group, which are skilled geneticists. If we do proteins, we do what we call proteomics. We measure with mass spectrometry, which I will tell you a bit of what that is. These complex protein mixtures that we have in a system. Or if we go for small metabolites, we call it metabolomics then. We use in my lab a lot of instruments called mass spectrometers. That's very expensive balances, you could say, that can monitor a delicate concentration of all different kind of molecules and tell exactly how the structure is and how much of the molecules there are in a complex molecular sample. Once you have the mass spectrometer, that doesn't solve everything. You have to also present your molecules in some delicate way to the instrument. So you have to get samples selected, for instance, body fluids that we can infuse into the instrument by a technique called electrospray, or we can produce small droplets of the sample on a solid surface and then do laser ablation, or we can present tissues directly into the instrument if we have material tissue that we can measure. We are pretty sensitive when we do this analysis, and that's the key. We want to measure things very accurately and at very high sensitivity. I've made a little calculation for you just to understand what sensitivity is when we talk about sensitivity. So let's say you take one piece of sugar and you put that in a cup of coffee, mix it carefully, and you will end up with a concentration of sugar in that cup of coffee approximately 0.03 mole per liter. And all of you remember Avogadro's number, I guess, from school. So if not, this is it. So 1 molar is 1 mole per liter, and that's 6 times 10 to the 23 molecules per liter then. We take that cup of coffee and then we bring it to our Olympic swimming pool next door, and we spill that cup of coffee in that swimming pool, and then we mix that carefully. It can take a while to mix it out carefully in that big volume. And then we will have 1 nanomolar concentration in that pool. Then we take another cup of coffee and get some water from the first pool and put that in the second pool and mix it carefully. And then we draw a sample from that. And then we get a concentration about 10 femtomolar, so 10 times 10 to the minus 15 mole per liter. And that is the sensitivity we can do. So we can measure how much sugar there is in that second swimming pool then. Unfortunately, swimming pools are a bit problematic because, you know, it's not only water and sugar in a swimming pool. Kids' pools, terrible. So we have problems that we have a matrix all the time. We have to fight background. So think about that next time you go to the swimming pool. Or maybe not. We do other things also. We don't only use mass spectrometers. We use targeted proteomics. And targeted proteomics is when we know what we are looking for. So we're not screening. We are just measuring things with our precise methods. And we have used many different ways, but one of the ways that I would tell you shortly about is proximity extension assays, you see at the lower panel here. And what you do then, you have 2 binders that can detect your molecule of interest, a protein, for instance. And once these 2 binders come close to each other, they have single-stranded DNA on there, conjugated on them, as you see here. And those can ligate, and this ligation then can be amplified by PCR, polymerase chain reaction. And this is a technique that has been developed in Uppsala by Olink, which is a company. And we use that, and we have used that in a number of studies where we also have included ME/CFS patients. And I mentioned this last year, and now we have concluded the data for the first Swedish population of ME/CFS. And the unique thing here is that we have both cerebrospinal fluid, which is my body fluid of interest, and plasma from these patients. And we also compare them with fibromyalgia patients, where we have CSF and healthy match controls. And we've been very, very careful with how we selected the material to make sure that we can draw conclusions. And that's extremely important, especially when we have small populations. So we run selectively neuroinflammatory markers, 92 in the first set. And just to conclude, what we have found is in cerebrospinal fluid, we find significantly enriched molecules that tells us that patients with both fibromyalgia and ME/CFS have a neuroinflammatory ongoing reaction that we can measure. Plasma samples, blood samples, did not reveal that much, actually. A bit of a disappointment to us. It would be much easier if we could measure these in blood, but unfortunately that's not given so much yet. We are now following up with a second study to validate our first findings, but also to see what else we can do. And we have now included a small group of very highly selected patients. We have ME patients, we have our multiple sclerosis contrast patient group, and then we have our healthy match controls. And we have focused only on cerebrospinal fluid. 7 of each is a very small number, but this is what we are going to do. We are going to set them into a totally new set of panels that have not been tested before for cerebrospinal fluid. And we have the luck to work together with Olink on this. So about 1,200 different selected markers will be selected for this. Based on the first 2 panels that we just got data from, actually already now we have 36 significantly differentiated cerebrospinal fluid related proteins that we can find in ME/CFS patients. This of course has to be validated then in a larger set. And we also see both similarities and differences between multiple sclerosis and ME, which is, I think is of great importance to understand this. With the proteins that we found initially is also following what we found earlier, which is very good. So we find neuroinflammation, cell damage, and repair systems ongoing in these patients, which is, of course, of great importance. Secondly, we do targeted steroidomics. We look at molecules that are of steroid family classes. And steroids are extremely important for controlling many different things in the body. So, for instance, reproduction, endocrinology, maturation of different cells, gene expression, neurological functions, etc. And we use steroids for classic endocrine detection in many diseases. But the trick is that in many studies, there's only 1 or 2 markers measured at one point. But we have a possibility now to do full screen, steroidomics or steroid hormone analysis. We can measure the whole biochemical pathway with our techniques. And what we've done in this first step, and this is also supported by Open Medicine Foundation and the Swedish patient organization, is that we have selected 24 patients and 24 match controls. And then we have done in-depth screening of all the steroids. And I will just give you a brief idea of how this is done. We can do it in plasma now. We don't need cerebrospinal fluid. We can do a derivatization and then a high-resolution separation using separation technique called supercritical fluid chromatography, and then high-resolution mass spectrometry to measure what we have. And what we found is maybe not dramatic changes, but we have changes that I think is relevant when we'll come to discussion what's going on in the patient group. So if I have a general downregulation of many of the steroids, the only one that came out significant, as you see here, is the pregnenolone. And I will show you what that can mean just soon. Pregnenolone is the key steroid in the biochemical pathway. So it's actually the precursor of the rest of the steroid profiles. And I also put these blue arrows in the biochemical pathway here. And all of these enzymatic ways that I marked here are located to a specific organelle. Is there anybody who can guess what organelle that is? Mitochondria. So pregnenolone is one of the neurotransmitters or neurosteroids that we have in fairly high concentrations in the brain. It controls, together with its sulfate product, synaptic function, neuroprotective functions, and also enhanced myelinization. And it can also, together with sulfite ester, improve cognitive and memory function. So if we have a general reduction of pregnenolone, we can tie that to some of the symptoms. And it is produced in mitochondria. So things start to tie together in a nice way here. Finally, what about autoimmunity then? Autoimmunity is something that we have been working with now for a while. That is when the body reacts with immune system reactivity against something of your own, so a protein or target of the body. And multiple sclerosis is one example of that autoimmunity disease. There are many different autoimmune diseases, around 80 chronic inflammatory diseases. Prevalence in the United States is up to 8%, and it's more common in women than in men. And for women, it's actually the 4th largest class of disease. And in ME/CFS patients, there have been studies, and there are ongoing studies also from other groups. And I started approaching a group in Berlin run by Carmen Scheibenbogen. And she had found in 2015 that she found in a subset of her patients with ME/CFS an upregulation of autoantibodies against muscarinic and beta-adrenergic receptors, which I found very interesting. So we asked for help, and we also selected a bit broader panel of autoimmune reactivity for our studies and for our Swedish patients to look at adrenergic receptors, or antibodies against them, and also against the muscarinic receptors here. And the biological function of adrenergic receptors and muscarinic receptors also tie to what we see in the patients as symptoms. So the study design was we took 48 patients with matched controls. We took both cerebrospinal fluid and plasma from these. And we could then verify and validate not only Carmen Scheibenbogen's findings, but also our own preliminary data. So we have a significant upregulation of autoantibodies against the beta-adrenergic receptors, as you see here, and also some of the muscarinic receptors. And this is a log scale plot here. So you see that in healthy controls and in patients, we have differences, but we have a big biological spread also, which is what we expect here. So everybody can carry some of these autoantibodies and still not have any symptoms, but significantly enriched in patients in this case for some of them. See if they move ahead here. Yes. What we also found was in cerebrospinal fluid, no autoantibodies for these muscarinic and beta-adrenergic receptors, which is something positive, I would say. So we have validated the findings, summarized here, significantly elevated in patients. We have observed now in 2 separate studies in Sweden, and we also have no detectable levels of autoantibodies in our cerebrospinal fluid, which is good. Final minute, just something that is not totally related to ME/CFS, but something that I think has given me at least some ideas of what to look at. We just published a study together with a lot of colleagues in Uppsala, where we have looked at acute sleep loss and in an experimental setup that I will tell you. And the remarkable change that you can see after just 24 hours of sleep deprivation. So the setup of the study briefly is that we have young male healthy individuals, students of course, that we select 15 of them, and we ask them not to sleep for 12 hours when they're supposed to sleep, basically. And we take blood samples, we take adipose tissue, fat tissue, and we take muscle biopsies. And then we do screening of all different kinds of molecules. And just to summarize, what we found is a dramatic change in genes, specifically those genes that are regulating our daily rhythm or circadian rhythm. And we also find a degradation of muscle tissue. We find an upregulation of adipose tissue. So what is that meaning? Well, after only 12 hours of depraved sleep disturbance, we have phenomena going on in the body. And I know that patients have very difficulties with their sleep. So, of course, this is something I want to follow up on and see what kind of metabolic changes and proteomics changes and maybe gene changes we can see. So, conclusion then. Significantly elevated levels of neuroinflammatory markers have been established, especially in cerebrospinal fluid, which is the target for me. Disturbed steroidogenesis in the patients, and it could be related to dysfunctional mitochondria, significantly elevated levels of these autoantibodies. And sleep hygiene is very important. That leads me into my final slide here. I had the chance to be on a tall ship race ship this summer for 2 weeks. We managed to sleep like 3 hours in a row for 2 weeks, which is, I can tell you, you get tired and dizzy, and I don't know if we didn't gain much fat, I think, because food on board wasn't the best, maybe, but that's. But we had also very tough weather. We had 8-9 Beauforts in this race, so that was a fantastic expedition, actually. They came second in this race with the ship in the end of the race. So with that, I would like to state what Lord Kelvin said at one time. When you're face to face with a difficulty, you are up against a discovery. And I think that's where we are now in this field. And I really look forward to follow what's going on in all the other research groups around the world that are focusing on this. So a great thank to Ron's family for arranging this, Linda with family, and all their involvement in this research field, to Open Medicine Foundation, of course, and to our Swedish patient organization who supports us, colleagues all over the world, and of course all patients and healthy controls that voluntarily give samples for this research. So thank you very much. Tack så mycket.